I'm Dr. Kiran Ramrao Borkar. Now today I'm going to deliver or discuss about the gluconeogenesis. Now, first of all, we see here what is gluconeogenesis. Gluconeogenesis is that the synthesis of glucose from non-carbohydrate compound is known as a gluconeogenesis. Okay, then what type of the non-carbohydrate compound? That is lactate, glycerol, glucogenic amino acid, propionic acid, intermediate of the TCA. All these are the non-carbohydrate substance which is used in gluconeogenesis. Okay. Now we see here the site of gluconeogenesis. The main site of gluconeogenesis is that the liver, then lesser extent in the renal cortex. The pathway is partly mitochondrial and partly cytoplasmic. Now, we see the substrate of gluconeogenesis. That is the first substrate we see here, the alanine. Okay, what happens here? Pyruvate is formed. Look at this. This pyruvate is formed from glycolysis in the muscles. Look at here, in the muscles. During the degradations of protein which occur during the starvation, this alanine is released into the blood and this alanine is released by muscle into the blood and then taken up by liver and converted back to pyruvate. Then converted back to the pyruvate by reverse transamination reaction that occur in muscle. Pyruvate is used to produce glucose. Via gluconeogenesis. Okay. Now we see the another substrate is that the glycerol. Okay. This glycerol is formed in adipose tissue by lipolysis of triacyl glycerol. It is also called as a triglyceride. This glycerol released into the blood. And taken up by the liver where it is phosphorylated to 3-phosphoglycerate, which is intermediate in gluconeogenesis. Okay. Now we see the another substrate of gluconeogenesis is that the lactate. Okay. This one is the Cori cycle. What happens in Cori cycle? This pyruvate is formed glucose by glycolysis in muscle cell and erythrocyte. This lactate is converted to erythrocyte is converted to lactate. This pyruvate is converted to lactate by LDH. What is LDH? Lactate dehydrogenase enzyme. This pyruvate, then this lactate released into the blood and taken up by the liver and converted pyruvate by LDH. Here also converted LDH and here also converted LDH. This pyruvate is converted to glucose. This pyruvate is converted to glucose via gluconeogenesis in liver. In liver and released into the blood. This glucose released into the blood where it can be used as an energy source for muscle as well as other tissue. Okay. Now we see here, look at here, a lactate. Here also the lactate form from the pyruvate but in erythrocyte or red blood cells. Here also this lactate released into the blood and then this lactate taken up by the liver and then this lactate converted into the pyruvate by LDH and then this pyruvate convert glucose by gluconeogenesis process. Okay. Now we see here the reaction of gluconeogenesis. Look at here. This is all about the this one is the glycolysis and this one is the gluconeogenesis. All these are the reactions of gluconeogenesis. Okay. The first reaction of gluconeogenesis is that the pyruvate to oxaloacetate. What happens here? The ATP hydrolysis provides energy for carboxylation of pyruvate. The reaction is catalyzed by the pyruvate carboxylase. Okay. And this reaction occurs in mitochondrial matrix. But this oxaloacetate cannot permeate the mitochondrial membrane well and must be transported across the membrane in the form of malate. Okay. Then this oxaloacetate to then second reaction is that the oxaloacetate to the phosphoenol pyruvate. In short, this phosphoenol pyruvate is also called as a PEP. What happens here? This reaction occurs in cytoplasm. Okay. The hydrolysis of GTP provides the energy for the carboxylation of oxaloacetate. Then this phosphoenol pyruvate to fructose 1,6-phosphate. Look at here. Fructose 1,6-bisphosphate. This conversion, that is from phosphoenol pyruvate to the fructose 1,6-bisphosphate, this conversion occurs by 6 sequential reactions that are simply the reverse those that occur in glycolysis and are catalyzed by the same enzyme, that is enolase, then phosphoglycerate mutase, phosphoglycerate kinase, and glyceraldehyde phosphate dehydrogenase and triose phosphate isomerase. All these enzymes use or catalyze all these 6 reactions and and this phosphoenol pyruvate get converted into the fructose 1,6-bisphosphate. Okay. Now we see another reaction of gluconeogenesis is that the fructose 1,6-bisphosphate to fructose 6-phosphate. This reaction is a reverse of reaction catalyzed by phosphofructokinase during the glycolysis. Activator of kinase is citrate. Now we see the next reaction of gluconeogenesis is that the fructose 6-phosphate to the glucose 6-phosphate. This is the reverse reaction of glycolysis. Here also some enzyme activate the reaction. Then which enzyme? Phosphohexose isomerase enzyme catalyze this reaction, and this fructose 6-phosphate get converted into the glucose 6-phosphate. Now we see the 6 or next reaction of gluconeogenesis is that the glucose 6-phosphate to glucose. The reverse reactions of glucokinase and hexokinase during the glycolysis. However, in gluconeogenesis, this reaction is catalyzed by glucose 6-phosphatase. This reaction is catalyzed by glucose 6-phosphatase, which is present only in gluconeogenic tissues. This enzyme present only in gluconeogenic tissue. Okay, this is all about the stepwise reaction of gluconeogenesis. At gluconeogenesis, it means that conversion of pyruvate to the glucose. Okay, now we see here the how this gluconeogenesis process regulate. It means that regulation, we see here the regulation of gluconeogenesis. Gluconeogenesis and glycolysis are reciprocally regulated. One pathway is relatively inactive when other is active. Regulatory enzyme. Then what type of the regulatory enzyme used here? Pyruvate carboxylase, fructose 1,6-bisphosphatase, then ATP. Then we see the hormonal regulation of gluconeogenesis. Glucagon and glucocorticoids increase gluconeogenesis. What happens here? This glucagon and glucocorticoids can stimulate the synthesis of enzyme of the gluconeogenic pathway, which was used to bypass irreversible step of glycolysis. Now, the another hormone, that is the glucocorticoids, induce the synthesis of hepatic and amino transferase and provide substrate for the gluconeogenesis. Okay. Now, look at here, this hormonal regulation of gluconeogenesis. This process is the glycolysis, and this one is the gluconeogenesis. In gluconeogenesis, glucagon and glucocorticoids increase. Then this gluconeogenesis process active here. Okay. Now, we see here the significance or importance of gluconeogenesis. Why the gluconeogenesis process important here? Why the glucose formation important here? Look at here. Brain and CNS, that is central nervous system, erythrocyte, testis and kidney, medulla are dependent on glucose for continuous supply for energy. This is the first importance of gluconeogenesis. Then, human brain alone require about 120 gram glucose per day out of 160 gram needed by the entire body. Glucose is the only source that supplies to the skeletal muscle anaerobic conditions, and this glucose formed from the gluconeogenesis. Now, during the starvation, gluconeogenesis maintains the blood glucose level. Another importance is that the stored glycogen is depleted within the first 12 to 18 hours of fasting. On prolonged starvation, the gluconeogenesis is speeded up and protein catabolism provides the substrate, namely glucogenic amino acids. All these are the importance of gluconeogenesis. Okay, now thank you so much.